Hi everyone, and thank you for joining us for BioTek's webinar, Kinetic Proliferation Assay Using High Contrast Bright Field Imaging. My name is Dylan. I'll be your moderator today. If you have any questions during the webinar, please submit them with the chat feature on the bottom left-hand corner of your screen. We'll do our best to get to as many questions as possible at the conclusion of the event. Today's webinar will be presented by Joe Clayton, Principal Scientist here at BioTek. We'd like to thank you all for joining us, and with that, I will turn things over to Joe.
Okay, thank you, Dylan, and thank you, everyone, for joining us online. Today's webinar is titled Kinetic Proliferation Assays Using High Contrast Bright Field. And the focus of this presentation is describing how BioTek live cell imaging systems can be used to conduct long-term automated cell proliferation studies. This will include a few examples that demonstrate the unique quantitative analysis that these kinetic assays support and the advantages that they have over single endpoint measures. And throughout this presentation, I'll highlight how this imaging-based approach enables detailed qualitative assessment of cell proliferation as well as changes in cell morphology over time. The content of this webinar is outlined here. I'll begin with some background on BioTek instruments and describe some of the key features of our automated live cell imagers. I'll then review existing approaches to measuring cell proliferation and the advantages of this kinetic imaging-based approach over conventional single endpoint measures. I'll then present two label-free methods for quantifying cell proliferation using high-contrast bright field imaging. The first method uses percent confluence as a readout for cell population growth over time. I'll outline the setup of this assay and the use of Gen 5 image analysis software to conveniently and objectively measure percent confluence over time and across multiple conditions. I'll then go over how this method can be used to quantify cellular growth response to anti-proliferation drugs or modifications to gene expression. I'll then introduce high-contrast bright field cell counting and describe how this powerful label-free method can accurately identify and count adherent cells over a broad range of cell densities. This method enables researchers to directly measure changes in cell number in real time without disrupting the cells with stains or having to deal with the effort and time required to achieve a stable line expressing fluorescent markers. And as with percent confluence, I'll describe the setup of a high-contrast bright field cell counting assay and go over how it can be used to generate kinetic proliferation profiles that enable detailed quantitative analysis of cell growth. And I'll present some data comparing high-contrast bright field cell counting to percent confluence measurements, as well as compare this label-free cell counting method to counts achieved in parallel using GFP labeled cells. And lastly, I'll briefly describe how these kinetic proliferation assays can be multiplexed with cytotoxicity or apoptosis markers for more detailed characterization of cell growth. For wrapping up with some conclusions at the end and some time for some questions. So at BioTek, we design and manufacture innovative life science instruments, including automated live cell imagers, microplate detection systems, liquid handlers and plate stackers, and the BioSpa, which is a fully automated incubation system. So today's presentation will focus on kinetic live cell imaging applications So I'll briefly describe the capabilities of our two imaging systems, the Citation imaging and multimode reader and the Lionheart FX automated live cell imager. All of the data that I'll present today was generated with a Citation 5 linked to a BioSpa system, although the proliferation assays that I'll describe can also be conducted by swapping in a Citation 1, and similar multi-day proliferation assays can also be run on the Lionheart FX. So looking a little closer at these imaging systems, both systems are available with fluorescence, phase contrast, bright field, color bright field, and high contrast bright field imaging, making these instruments extremely versatile for a range of applications. Additionally, both the Citation and Lionheart FX have integrated temperature control and available CO2 and O2 regulation, as well as a humidity chamber that is available on the Lionheart FX. For fluorescence imaging applications, the instrument can hold up to 4 imaging filter cubes, each containing a colored LED for excitation and filter set. Both of these instruments can hold up to 6 objectives in a turret, as shown here, ranging from 1.25 to 60x air objectives for the Citation, and 1.25 to 100x for the Lionheart FX as well, including a 60 and 100x oil immersion objective. In addition to user-trained autofocus, laser autofocus is also available on these instruments. And these are fully automated imagers, so the imaging channels, the magnification, exposure settings, and imaging wells can all be selected prior to the start of the run, enabling fully automated walk-away, single-point, or kinetic live cell imaging. Additionally, available dual reagent injectors and up to 20 frame per second image capture rates enable the characterization of rapid cellular events, such as calcium mobilization in response to GPCR activation. And on the other end of the time scale, these imagers are optimized to conduct long-term live cell studies. So in addition to the integrated environmental controls that I mentioned earlier, coupling the Citation with the BioSpa 8 enables complete environmental control and automation for experiments running hours up to weeks at a time. BioSpa accommodates up to 8 plates with the ability to run different experiments on each of those plates. And a liquid handling dispenser could be integrated into this system for complete workflow automation. So given these unique capabilities, these automated live cell imagers can be used for a wide range of applications, with just a few listed here. However, for the remainder of this presentation, we'll focus in on just one application in particular, which is cell proliferation. So, as you probably know, cell proliferation is defined as an increase in the number of cells in a culture or cell population, and it's really the balance between cell division and cell death, and in some cases differentiation, over a period of time. The proliferation studies provide valuable insight into both normal developmental processes as well as the characterization of disease models and treatments, in particular for cancer research. So measuring cell proliferation has conventionally relied on biochemical approaches, typically using either methods that are designed to stain and quantify DNA content or detection of cellular metabolic activity of some kind. So in addition to being quite labor-intensive, these indirect methods are often limited by artifacts, and they're difficult to normalize across conditions. And because these are endpoint assays, they're really not able or designed to be able to effectively follow changes in cell number over time, which is really the principal goal of a proliferation study. So with an endpoint proliferation assay, typically cells are plated and treatments are administered. Cells are then cultured for some amount of time before stains or reagents are added to measure bulk fluorescence of the population, or the cells are trypsinized and collected from the plate so that they can be analyzed or lysed and analyzed. So in the end, a single point in time is represented by these approaches. Or at best, multiple experiments have to be run out to different endpoints, and an attempt is then made to stitch these experiments together. Clearly not ideal. In contrast, kinetic imaging-based assay provides continuous monitoring of changes in cell population size, with high-resolution images being captured at regular intervals throughout the experiment. So this enables detailed analysis of cell proliferation over time and produces robust kinetic profiles. And because these reads are done continuously, you have the option of multiple treatments of the cells and monitoring those treatment effects over time. Now, one effective metric used to measure the size of an imaged population of cells is percent confluence. This is the proportion of the surface covered by cells. However, assessment of confluence is typically a very subjective evaluation. For instance, I imagine that if I was to ask you, all of us would agree that the cell confluency is different across these four images shown here. But if I was to ask each of you to quantify the specific confluence of these images, I would get a considerable range of values. So really, to get accurate and reproducible confluence measurements requires a rigorous and standardized image analysis method. And this is exactly what we get with this imaging system. So using Gen 5 image analysis software and high contrast bright field imaging, percent confluence can be determined routinely for long-term kinetic cell proliferation studies. This convenient, label-free method saves considerable time and expense, and importantly, it provides reproducible results for meaningful quantitative analysis. So it really takes the subjectivity out of the equation. And this method enables detailed characterization of cell morphology throughout the experiment, providing unique insight into changes in the biology over time. All right, so if we look at the setup for this multi-day kinetic proliferation assay, it consists of plating the cells at low density in a 96 or 384 well microplate. Cells are then treated with test compounds or anti-proliferation drugs. And then I think based on this microplate design, this format works quite well to simultaneously test multiple cell types or backgrounds, as well as testing the effect of transient gene knockdowns. So prepared plates are loaded into a BioSpa 8 automated incubator, and the kinetic high contrast imaging protocol is selected, which starts the assay. Plates are then routinely delivered to the Cytation imager from the BioSpa, and image analysis settings are applied while the experiment is running. So it's a very efficient and automated process from beginning to end. And if desired, media can be changed and additional treatments performed throughout the experiment. If we look at the image analysis settings in Gen 5, these need to be determined just once per cell type, and they're achieved using intuitive tools that achieve really accurate confluence measurements that are consistent and reproducible across a wide range of conditions. So most optimization of these settings is done by adjusting the threshold and object size, and this ensures that all cells are included in the analysis while excluding debris. Additionally, users have the ability to easily make adjustments through a range of advanced detection options to further refine the analysis. Now, determining the optimal image analysis settings are easily confirmed in this system by a real-time display of the resulting object masks that are placed around the cells, as seen here. So you can clearly identify when a threshold value is too high, as on the left, where you'll exclude some of the cells from the analysis, while threshold values that are set too low will quickly be revealed by including areas not occupied by cells. So you can easily visually confirm optimal settings at every stage. Doing this just for a few selected images from the initial experiment allows users to quickly determine optimal values, which are then consistently applied to the entire experiment. And importantly, these settings will be saved within the imaging protocol so that they can be automatically applied to any future experiments using that cell type or similar ones. So as an example of how percent confluence can be used to characterize cell proliferation, we have HT1080 cells that were seeded onto a 96-well microplate at 2,000 cells per well in this case, and treated with serial dilutions of two known anti-proliferation compounds, doxorubicin and circosporamide. So under these conditions, this experiment was run over 3 days with images collected every 2 hours. Now, as seen in this video, untreated HT1080 cells exhibit robust proliferation within this controlled environment, and the collected images provide a very detailed record of the entire experiment for future reference. And the automated image analysis that is applied across the entire microplate creates a profile of cell confluence over time for each well. And so that's what we're seeing here, a matrix view of the entire plate with each of the conditions displayed. So you'll see that this microplate format enables really efficient testing of multiple treatments or cell types with very consistent results across replicates. Now, if we compare the confluence profiles for each condition, as we see in this figure here, comparing multiple doxorubicin concentrations, this provides a very informative and intuitive readout of the treatment response corresponding to dosage, but also it really gives you a clear understanding of how the response varies over time. And this allows you to determine optimal time points for analysis retrospectively rather than being restricted to a single unoptimized predetermined endpoint at the onset. Additionally, measuring the area under the curve, or AUC, for each of these profiles enables the incorporation of treatment effect over time into the quantitative analysis. AUC values are generated within Gen 5, and these provide a unique method for comparing proliferation rates across treatments. And from these results, biologically relevant dose-response curves are generated, and right within the Gen 5 software. And this enables quantitative quantification of drug potency, such as EC50 values, done very conveniently and in a very unique, providing very unique information about the treatment. Now, images that have been automatically collected and analyzed throughout the proliferation experiment can be reviewed, providing valuable insight into the biological process and to evaluate specific time points and results of interest. So, for instance, we might be curious about the relatively weak inhibition seen at low concentrations of doxorubicin, leading us to focus in on the time point highlighted here in red, 48 hours into the experiment. So here the mean confluency of negative control wells, that's those treated with vehicle alone, shown in gray, is 97%. Whereas the mean confluence of wells treated with 10 nanomolar doxorubicin, shown in light blue, is 85%. So a small but significant decrease in the proliferation is seen. However, when we review the corresponding images shown for these conditions at this time point, we observe that there appears to be much more to the story. Although there's an inhibitory effect caused by the doxorubicin treatment that is clearly detected by the differences in confluency, closer inspection shows that there appears to be considerably less cells present after 48 hours in the treatment condition than the confluence values might indicate. So with the doxorubicin treatment, cells appear larger and more spread out compared to the negative control. So this means that the actual inhibitory effect on cell growth, on the overall cell population size caused by this treatment is likely even greater than is represented by the 12% or so difference in confluence. So therefore, these images provide not only a window into the underlying biology, but it also allows researchers to identify potentially confounding conditions, providing greater confidence in the assay results compared to alternative methods that are less transparent. However, keeping in mind that the number of cells in a population is what all proliferation metrics are really intended to address, this example demonstrates the importance of a much broader question, which is, what is the relationship between percent confluence and actual cell number? And how does this relationship change at different cell population densities? So to answer this question, we need a way to identify and count cells directly, which we can do by labeling each cell with a nuclear stain like Hoechst and comparing cell counts at varying cell densities to corresponding confluence measurements. So if we set up a cell titration plate containing a wide range of cell numbers loaded per well that are then stained with Hoechst dye, we can address this. So each well is imaged with high contrast bright field illumination for confluence measurements, as well as fluorescence illumination to view the stained nuclei, which are readily identified and counted by Gen 5 software as shown in the images on the right. So if we take the ratio of these two measurements, percent confluence versus these labeled nuclei cell counts shown in this graph, it reveals that confluence values often do not have a linear relationship with cell number. And this is especially true at higher cell densities for cell types that are not contact inhibited, like these HT1080s. So given this, for many applications, direct cell counts clearly would be the preferred method for determining the size of a cell population and for measuring changes in the population size over time. However, nuclear stains are known to be potent disruptors of cell division and therefore don't support kinetic studies of cell proliferation. Alternatively, approaches requiring the generation of cell lines that stably express fluorescent nuclear tags are labor-intensive and limited by expensive reagents with poor transduction efficiencies often. So ideally, a label-free method could be used to conduct kinetic cell proliferation assays. So for this reason, we've developed a convenient and automated label-free method for cell counting using high-contrast bright-field imaging. This method is illustrated in this panel of images, in which each of the cells shown in detail in the first image on the left can be made to produce a discrete bright spot of light among a relatively or almost black background seen in the second image. Now this effect is achieved by the dramatic contrast enhancing properties of the high contrast bright field imaging technique. And if we overlay these images as seen in this third image, it demonstrates the effectiveness of this label-free technique, which also enables then Gen 5 image analysis to conveniently identify and count each cell as illustrated in the image in the far right. And this graph shows that the ratio of cell counts generated using this high contrast bright field method with counts achieved using nuclear stains, as described on the previous slide, confirms that this label-free method produces accurate cell counts across a wide range of cell densities. All right, so returning to this assay setup slide, you see that just as with percent confluence, the setup is really quite simple. In fact, with the same setup and a single protocol, an experiment utilizing both percent confluence measurements and direct cell counts can be performed, giving maximum flexibility to analyze your data. And again, once this assay is started, plates can be accessed during any interval between reads if needed, or the plates can be completely left alone with all necessary environmental controls maintained by the instruments. And I didn't mention this before, but the coupled BioSpa 8 and Citation 5 were designed to fit conveniently within a biosafety cabinet. However, because many applications will never require removal of the microplate lids during the experiment, which is the case for these proliferation assays, you may prefer to keep the system right out on the benchtop, which is how all of these experiments were run. All right, so now let's run through the same 3-day kinetic proliferation assay, only now using high contrast bright field cell counting as our readout. Looking at this video of this experiment, we'll see, you can see that as this population grows, this label-free method allows us to effectively resolve each cell over time so that we can generate accurate cell counts all the way up to full confluence. And looking at a matrix view of the entire 96-well plate, we see that this technique produces highly consistent proliferation profiles across all replicates for really robust and reproducible results. And if we compare the kinetic profiles of cell counts from a selection of doxorubicin concentrations, just as before, versus control cells, this illustrates just how sensitive this method is at detecting differences in proliferation rates. As before, we can use the area under the curve for each cell count to quantitatively compare the proliferation rates across conditions using cell counts. And then from these generate very informative dose-response curves that really provide increased sensitivity with more direct approach to the evaluation. This is really illustrated when we compare the percent confluence results to our direct cell counts. We see that the conclusions we would draw from both are pretty much the same. However, the direct cell counting experiment is much more sensitive at picking up inhibition by doxorubicin in this case. And we see this reflected by the 8 to 9-fold lower doxorubicin EC50 value that we get when using the direct cell counting method to measure proliferation. Now, given this, it seems clear that when available, a method that relies on direct cell counts would be preferable over confluence as a means of measuring proliferation. So we wanted to see how this label-free cell counting method compared to counts of fluorescently labeled nuclei side by side in a kinetic proliferation assay. So to do that, we ran the same 3-day kinetic proliferation experiment using an HCT116 cell line that stably expresses the nuclear marker H2B GFP. So each of these cells has a bright GFP labeled nucleus. And we image these cells every 2 hours, just as before, using both high contrast bright field as well as now fluorescence illumination. We then conducted cell counts in Gen 5 using both sets of images. Now you'll see the resulting counts using these two techniques were highly similar and generated comparable kinetic profiles. And this is with a cell type that tends to cluster together, which actually makes it resolving these individual cells even more difficult than we would see with cell types that tend to distribute themselves more evenly. And even more telling, the dose-response curve and calculated EC50 value generated using this label-free method is effectively the same as those generated from the fluorescent label-based approach. So you get the sensitivity and accuracy of counting cells directly with the convenience and practicality of not having to deal with fluorescent labels. And lastly, with a wide selection of available image filter cubes and 4 filter cube slots in the Citation and Lionheart FX imagers, these kinetic cell proliferation assays can be combined with multiple reagents for measuring cytotoxicity, necrosis, or apoptosis. As an example, we added CellTox Green, which is a cell membrane integrity marker used for measuring cytotoxicity. So we added this to a proliferation experiment in which HT1080 cells were treated with doxorubicin. In the panel of images on the left, it illustrates how, with increasing concentrations of doxorubicin, a corresponding increase in CellTox Green fluorescence is observed. And changes in CellTox Green fluorescence over time can be seen in this video of cells treated, in this case, with 100 nanomolar doxorubicin. So now, total CellTox Green fluorescence can be quantified over time relative to proliferation profiles. Alternatively, counts of CellTox Green positive cells can be generated and then reported relative to percent confluence, or as a proportion of the total number of cells, as shown in the bottom graph here, allowing you to more effectively normalize results across conditions. And dose response curves can be generated from these experiments to add yet another dimension to the characterization of anti-proliferation compounds for these studies. So to wrap up this presentation, I'd like to just summarize a couple of the main themes. Firstly, kinetic cell proliferation assays really enable detailed and unique analysis of cell growth and viability, and they offer a range of benefits over single point and endpoint assays. High contrast bright field imaging is a label-free method for measuring proliferation that enables insightful quantitative as well as qualitative evaluation of cellular response. And this automated system provides a convenient and reproducible method for conducting robust proliferation assays. And proliferation rates can be measured using percent confluence, or label-free direct cell counts can be used to provide an even more sensitive and straightforward evaluation of cell proliferation. And so with that, I'll turn it back over to Dylan, and we can take some questions at the end.
Thank you, Joe. That was great. If you guys have any questions outstanding, please leave them in the chat window here to the left. And Joe's going to take a quick look through these and try and answer as many as we can.
Okay, so there's a question about the Citation 3. Wondering if the Citation 3 can be used to run these proliferation experiments. And the answer is absolutely. So the Citation 3 can be used and outfitted with the high contrast bright field kit and coupled with a BioSpa 8 incubator to run effectively the same assays. Yeah, absolutely. So a question here about using this technique on different plates. So for these assays, we used 96 well clear bottom, flat clear bottom tissue culture treated plates that with black sides. There are other plates that can be used, although I'd refer or recommend you visit our website and some of our application notes to really get, I think, a more detailed answer on that, exactly which plate types work well for this assay. But it is quite versatile, and it doesn't require specific plates. We have a question about high contrast bright field being used for a variety of cell lines. And this is an important question. High contrast bright field imaging really provides detailed imaging for all cell types. Some cell types certainly behave better as far as spreading out and being accurately counted. We've tested this on a variety of cells, and they've all worked quite well. I'd say the basic limitation is with cell types that tend to cluster. And of course, the same limitations in measuring those cells exist with alternative methods as well. So really the same limitations that you see with fluorescent-based approaches are going to be the same ones that are present here, but it really works across a variety of cell types. And really the power is that you can optimize the analysis for each cell type. The question about how are the counts achieved. So all these counts are done automatically through Gen 5. So you get readouts of the cell number in real time, and those are all compiled, essentially just counting the number of objects in the field. And those are compiled over time, and those are what are used to either have a readout of cell number at any given time or for the kinetic analysis. So yeah, that's completely automated. So is the resolution high enough to count bacteria cells using bright field application? So you can certainly pick up, depending on the bacteria, you can certainly pick up bacteria. Depending on the exact setup there, there might be varying degrees of effectiveness with extremely small objects. But it has been tested on smaller cell types as well. Okay, could you describe the label-free cell counting process with more detail? So the basic setup of this technique is it uses an aperture and a modified focal height to really get maximum contrast from these images. That can be through processing within Gen 5, resolved into individual discrete signals for each cell for accurate cell counts. It's really a combination of the high contrast bright field kit and the Gen 5 analysis. So, question about taking the images and then analyzing the cell number by itself. So this is kind of similar to the previous question. This is all automated. Really, the user defines the analysis settings at the beginning, and then all those settings are applied uniformly and routinely across all images automatically, and that's actually done while can be done while the experiment is running. So it really, when the end of the experiment comes up, it's the analysis is in most cases already done. So a question about focusing on floating objects or debris instead of cells at the bottom. So I would recommend using autofocusing techniques, laser autofocus techniques, ideally to get really accurate, consistent focusing of these cells. And that's what I've done in the experiments described here. And in those cases, the system is able to very effectively avoid focusing on debris, really producing consistent in-focus images throughout the experiments. A question about the software, the Gen 5 software and the analysis. A lot of questions about the automation of this, I think is a common theme. And it's really an important point is that this is a truly automated system where once you set up the imaging and the analysis for your conditions and determine what you want as readouts and what kind of analysis you want to have done, Gen 5 software automatically does all of that for you. So all those calculations are done behind the scenes. You can certainly have all access to what's going on there, but it's all done for you, including the proliferation profiles, these kinetic profiles that I've shown, and the area under the curve values that I think is a great way to quantify the effects. So yeah, so these are all done. It's a fully automated analysis that the user has the ability to control or to at least set up the analysis based on each condition. So for the high contrast bright field imaging, there's a question about what is required for that. And so I covered this a little bit already, but it's really the high contrast bright field kit and either the Lionheart FX or the Citation imagers can accommodate those kits. And that's really all you need to do this. That's the only specific components required, and everything else is just conventional or standard labware. The cells do need to be adhered to the surface. So this technique really is limited to adherent cells. So alternative methods would be required to quantitate, quantify counts of cells in suspension. You really need a monolayer of cells that can be focused on. Could this be useful for scratch assays during studying migration? Absolutely. So high contrast bright field works very well for migration assays, for scratch assays in particular. And I would direct you to some app notes we currently have in development that will be available online shortly. But this technique works very well to characterize cell migration in treatments for a variety of cell types. Let me just see if we have some other questions I've covered. I think a lot of these already. A question about the analysis, particularly using these other reagents. So the ability to exclude cells from your counts or include them based on markers, and you absolutely have this option. So you can use multiple masking. You can use dual masking on your cells. You can form a mask around cells using one channel, such as your high contrast bright field, and then use a secondary mask to look at, to measure reagent, a fluorescent reagent, and you can actually set thresholds for your analysis to exclude or include cells based on the research, whatever the biology really dictates. Does this technique require, so I covered this a little bit, so it really doesn't require specialized plates. There are certain requirements. You do want an imaging grade microplate to really get good resolution, but there are a variety of options available for that. A question about the high contrast bright field replacing phase contrast. So these are actually, you can run both of these at the same time. So you could actually run a proliferation experiment in which you use high contrast bright field on some of the wells or some of the plates and phase contrast for others. So you really have a whole lot of flexibility as far as what you want to analyze for a given experiment. So the high contrast bright field really does not exclude the option to use other imaging methods within the same experiment. It's really just you're only limited by the objectives that you have in your instrument. All right, so we'll take just one more question here. So a question about regular bright field versus the high contrast technique that we've described here. And so, of course, the problem with regular bright field is that it inherently has very poor contrast. So it's very difficult to quantify images, although it certainly can be done with various techniques. But the high contrast bright field kit really gives you maximum resolution and detail of your cells compared to bright field, and it's critical for this high contrast bright field cell counting technique that we've described here.
Great. Thank you, Joe. And thank you, everybody, for attending. We have a number of other questions on here that we will do our best to get back to offline. We have everybody's emails, so we should be able to take care of that. But on behalf of BioTek Instruments, we'd like to thank you all for joining us today. You will receive this recorded event and a transcript. For more information on BioTek's products and applications, we encourage you to visit our website, www.biotek.com. Be sure to check out our blog, Scientifically Speaking, and connect with us on Facebook and Twitter. Thank you again for joining us, and thank you to our presenter, Joe Clayton.